would like to start by thanking you for this kind invitation. And I'm here representing not only my laboratory and the research we have been doing, but also the European Reference Network, Genturis, that is operating now in Europe to standardize care for patients across Europe and across all European countries. So my main area of research is gastric cancer, and it's hereditary diffuse gastric cancer. So as we heard before, most of the gastric cancers, they occur in a sporadic form, and only 10% of them have a familial pattern. And some of those that have a familial pattern, they may be hereditary indeed. This means that they can be passed from parents to their offspring. And they are genetically determined from birth. And this means that you are born with a defect that predisposes you to develop cancer earlier in life than it would be expected for a sporadic situation. What is the important thing here is that the hereditary cases, because they are caused by a mutation that is in your DNA, we can identify it before a patient develops disease. So in fact, this is the only way we have to spot individuals at high risk prior to disease development. And this also gives the title to my talk. The bad thing is that from these 10%, we only know the reason or the defect, the genetic defect, in nearly 1 to 3% of all cancers. And in these people, they will have high risk of disease, and they will have a high penetrance genetic susceptibility, which is a mutation that is in the DNA. So I would like to present you the three main syndromes that we know in gastric cancer. The best well-known is hereditary diffuse gastric cancer. We call it HDGC in short. And it has been said already, it's characterized by these isolated cells that are non-adherent to each other, and they hide below a normal foveal epithelia, kind of hampering the detection sometimes by regular endoscopies. And this has been defined by CDH1 germline mutations, and CDH1 germline mutations are the main cause, the principal cause in the families that have this disease. Then we have another syndrome, it's called CAPS in short, it's a polyposis of the proximal stomach. And this disease is caused by a mutation, a point mutation that occurs in the promoter B of APC. And APC is known by promoting a different disease in the colorectal, which is called FAP. But in this particular situation, by targeting this promoter of this gene, is a different area of the same gene. It has a tropism to the stomach rather than to the colorectal. And then there is another syndrome that is well recognized, but does not have a genetic risk that we know of, that is familial intestinal gastric cancer, that is vastly understudied. So I'm going to tell you a little bit of what was the story of this syndrome, so you understand the difficulties we have, but also the benefits that everybody got because we found the germline and we can now follow up on patients. So the first discovery, I call it discovery 1, are germline mutations that are inherited in gastric cancer. And this is a discovery from 1998 from a New Zealand team, from Barry Guilford and colleagues, where they found that 3 Maori families, these are large families, that showed with early onset diffuse gastric cancer at 21 years old or 17 or 27. All these people have died of diffuse gastric cancer at very early in life, and this called the attention of the researchers. And in fact, 2 years ago, I've been in New Zealand for the 20th anniversary of this discovery. And actually the person that triggered all the patient that triggered all these discoveries who are there, and it was a beautiful experience. And so it took from 11 years until we got the next mechanism in hereditary diffuse gastric cancer. And this was occurring in 6 families with early onset diffuse gastric cancer. And these were germline deletions that were occurring exactly in the same gene, but this mechanism is so different that our diagnostic tools were not able to catch it. But at this time, we worked together with a company that developed the tool that now allows deletions in this gene to be discovered, and this has been introduced in clinical practice immediately after our discovery. Then, but you need to understand why does a gene actually cause disease, and this is what we study every day. So we need to understand what does a gene or a protein do that triggers the cancer, and this helps us to define the cause. So if you imagine that this is a normal epithelium, and in the epithelia, you have a monolayer of epithelial cells that are all attached to each other, right? You are born with one defect. And for every single gene in our body, we have two copies, one that we inherit from the father, one that we inherited from the mother. So if we are born with one, imagine that we inherit from our father that has a mutation in this gene, CDH1, we are at risk of developing disease. And this is what I mean with these symbols here. Now imagine that during your life, your stomach cells are attacked by something else, another mechanism, an aggression. And this causes what we call a second hit. And when that occurs, it means that the other allele, the one that we inherit from our mother, because the one we inherit from our father was already dysfunctional, also gets dysfunctional. And then we get these cells that are isolated and they are able to migrate, to invade the stomach and create this cancer that we call diffuse gastric cancer. But there are a lot of mutations that have been found, that genetic laboratories work hard to try to find the cause in many families. But in fact, not all the germline variants are disease causing. And this is a problem because you can't remove stomachs or do prophylaxis or treatments in people that actually do not have variants that are really causative of the disease. So we need to prove what are the CDH1 germline variants that we carry that are causative. And because of that, there was the creation of criteria to define causality. I am not going to bother you with this, but as patients, it's very important that you are able to understand what you read in reports. And if you are suspected of having a familial history of an hereditary disease, you should be very careful in looking of what is written, what is the classification that is given to the variant that is that you carry in your DNA. And the variants that are actually causative or actionable, as we like to call them, because you can do something to the patients when you find these variants, they need to be called likely pathogenic or pathogenic, because in this case, we have more than 90% of certainty that these variants will actually be the cause of disease. And this is not the case for many, many things. So the other thing that we need to know is if you have, imagine an inherited predisposition to develop this disease, you need to know what cancers will you develop, which cancers are you at risk to, because you need to surveil those organs prior to the development of the disease. So we need to study what is the disease spectrum, meaning what are the organs that are targeted by cancer when you carry a germline mutation. And it took us years to discover this. The initial families, they only had diffuse gastric cancer at an early age. That's why we call it the syndrome hereditary diffuse gastric cancer. But then we noticed that we also have colorectal cancers and lobular breast cancers at very early age. Then we found families that only had lobular breast cancer and did not have any gastric cancer. So the name was already wrong at that time, but we weren't sure. And then we found people that did not report any familial history, but they had an isolated case of diffuse gastric cancer at very early age, for instance, at 33, like I showed you. Or somebody that has two cancers, diffuse gastric cancer and lobular breast cancer, which are actually the two main cancer types that show up when you have this alteration. So these studies, and there were many, allowed us to define what are the cancers that occur when you have an actionable variant in the gene CDH1. And this is because these variants cause multifocal diffuse gastric cancer in the stomach, and this is transversal in many, many families. And in both genders, we can state that both genders develop recurrently and specifically multifocal diffuse gastric cancer, because this is the most clear evidence among these families. But then we have the lobular breast cancer. We have also certainty that lobular breast cancer is the specific histological type that you develop in this disease, not other types of breast cancer like ductal breast cancer or others. It's specifically lobular breast cancer, and we know that males are not affected. So if you are a carrier of a CDH1 variant and you inherit it from your parents and eventually will be able to transmit it to your offspring, you should be surveilled for breast cancer in female, if you are a female, but not if you are a male. Then there are other things that call our attention. So we were also quite glad to discover that in some families that carry CDH1 variants, you also find malformations. So this like cleft lip palate or aplasia cutis, as we call it. And these sometimes occur in patients that are from families, but they do not have cancer, right? But they are an indication that if you have a history of diffuse gastric cancer in your family, and also these malformations, it's another evidence that you may have a CDH1 variant, and you should be careful about that and request your consultation at the oncogenetics, an oncogenetic consultation at the hospital. So there are other germline defects in hereditary diffuse gastric cancer. So it's not only CDH1, and it was not until 2013 where a second gene was discovered. It's called CTNNA1. And this gene, it was found in one family, but now we know many families. So not as many as CDH1, but a dozen of families that carry mutations in this gene. And in this case, we know that this gene is important because it gives rise to tumors that are very, very similar to the ones that we see in the CDH1, and they are also multifocal in the stomach. So far, we do not have evidence that lobular breast cancer occurs when you have a CTNNA1 variant. So we may say that, as with the data that we have today, we know that if you are a carrier of a CTNNA1 mutation, you would be predisposed to diffuse type gastric cancer, but likely not to lobular breast cancer, unless your family has a history of that. So that the spectrum of hereditary diffuse gastric cancer is diffuse gastric cancer for CDH1 and CTNNA1. If cleft lip palate is only seen in families with a CDH1 variant, and lobular breast cancer as well. So what are the risks if you are a carrier? So if you are at risk for gastric cancer by the age of 80, in 42 to 70% in male carriers and 33 to 56% in female carriers. And this means that there is a high likelihood of you developing cancer. And this age of 80 is the age that we need to consider for this kind of studies. But many of these cancers that they develop early in life, in the 20s or 30s, that's why you need surveillance to be exhaustive. And the risk of breast cancer is 39 to 55% in female carriers, depending on the studies and the cohorts. So what can you do? So in a symptomatic carrier, so you know that you carry a mutation, you have no symptoms or disease, but you can do pre-symptomatic testing to see if you're a carrier. And after you identify the predisposing variant, you can undergo prevention. And prevention is done with intensive gastric surveillance for the stomach and intensive breast surveillance and risk reduction surgery. So in terms of how this should proceed, how we should proceed. So you should identify the patients at risk with HDGC based on the clinical criteria of the disease. You need that the genetic testing identifies a germline causative mutation. And then if you are positive, you need to be integrated in a program for risk assessment and application of risk reduction. That will be, if you have a CDH1 variant, applied to the stomach and the breasts, as I told you, with risk reduction surgery, which means removal of the stomach or removal at the breasts, knowing that the lobular breast cancer is detectable by MRI. And if you are a carrier of a CTNNA1, you should be surveilled for diffuse gastric cancer and have a risk reduction for surgery in your 20s or 30s. The lobular breast cancer needs to be evaluated on a case-by-case basis because I told you that we do not have evidence that lobular breast cancer may be a cancer that occurs in these families. So there are a lot of work put into the testing criteria, how to define clinical criteria, how do we know that the family have criteria for testing. And this has been an evolution. I just put it here in this slide that so you see that this is the criteria were only 2, and now we have plenty of criteria because of the diversity of the clinical presentations that we see in carriers. And then you can, so my presentation will be available. You are very welcome to look at it in detail, but it's different presentations of diffuse and lobular breast cancer, either alone or together. And very recently, we defined a new hereditary lobular breast cancer that is caused also by CDH1 and is present in families that do not present gastric cancer. And you can read, if you want, our paper that has all this very well explained that was published last August in Lancet Oncology. So as a summary, we have a population incidence of HDGC of 5 to 10 per 100,000 newborns, target stomach and breast. The histological types need to be very well defined. They are diffuse or lobular. It's a high mortality cancer for if you have a diffuse type cancer, diffuse gastric cancer, and moderate for lobular breast cancer. It's caused by the truncating alterations. So these are alterations that disrupt the function of these proteins. We know the defects that cause it. The penetrance is incomplete, which means that in the same family, you can have carriers that develop this disease and carriers that do not develop disease. We know the estimated risks, and you can have the presence, the fraction of families that carry these CDH1 variants with clinical criteria varies a lot depending on geographical origin. And if you are from a low incidence gastric cancer, you'll have a higher probability of finding a CDH1 than if you are from a country with great incidence of gastric cancer, because the families will be hidden among many sporadic cases. And then we have a second gene that is responsible for a very few families. And we know what to do in terms of prophylactics and treatment for the CDH1. And we are doing a lot of efforts for the CKN1 as well. But we need to solve 60 to 90% of HDGC families, which is a lot. And so there is a lot of things to do for the researchers like ourselves. So there is another, I don't know, I'm doing with time. Do I have another 5 minutes?
I suggest that you wrap up if possible.
Okay, so this is just a very short summary about GAPS. GAPS is the other syndrome, and I told you that it's caused by APC promoter mutations. It's caused by polyps rather than other perineoplastic lesions, and they are restricted to the proximal stomach, and they are in large numbers, and they have a family inheritance as well. And sometimes you can see some duodenal, or you can see colorectal cancers, colorectal polyps, although these were not described in the initial families. So I just want to finish by showing you something that we did to convince the national health system that is important to test people and to identify families. And this was by calculating the cost of following up families. So we followed over 70 individuals from the same family, and we proved that the costs related to people that are affected with cancer, this means that we allow cancers to develop in a family that has a mutation, this costs 100 times more than a negative patient that only costs the test. And if it's negative, goes home. If you are sick, you cost 100 times more. But if you are caught in an asymptomatic situation where you have a variant, but you still did not have cancer, you cost 10 times less than if you are sick. And this is actually very important to convince that prevention is important, and prevention saves lives and saves money as well. So I would like to finish without thanking all the team and the collaborators from my team and other institutes that have worked with us for many years, and the funding agencies. Thank you.